Hello, I'm Phoebe Lewis, a supplemental instructor for Dr. Byron Quinn's Natural Science Biology 1 course at Langston University, and I've compiled a series of review videos that will help you with your final. This particular one focuses on Chapter 8, which covers photosynthesis. So in this video, we're going to discuss the organisms that use photosynthesis, properties of light, the sections of the leaf, the light-dependent stage of photosynthesis, the Calvin cycle, an overview of photosynthesis in general, and photorespiration. So first, let's discuss the organisms that are able to use photosynthesis. And just a reminder, photosynthesis is the process in which, in layman terms, organisms are able to use energy from the sun to make their own foods, and particularly their own sugars. And so plants, as we know, algae, and cyanobacteria, this is a special type of bacteria, it's sometimes called blue-green bacteria, are able to use the sunlight to make their own food in the process of photosynthesis. So just the important thing to take from here, it's not just plants. Algae and some bacteria, specialized bacteria called cyanobacteria or blue-green bacteria, are able to are also able to use photosynthesis. So let's discuss the properties of light. Remember that sunlight has all of these different colors in it. This is the visible spectrum of light. And so sunlight actually has all of these colors in it. If you use a prism, you're able to see them. Rainbows are basically a natural prism that occurs. You're able to see all the different colors of this visible light spectrum. Well, let's talk about photosynthesis. We're talking about not all colors of light are used. It's mainly the blue and the red colors of light that power photosynthesis. Because if you remember, the plant leaf appears green because the green light is being scattered back to your eyes. Now all the other colors of light are absorbed, and the green color of light is being scattered to your eyes. That is why it appears green. The leaf appears green. Now that's an important concept. Think about black shirts and white shirts when you're going outside in the heat. The color black absorbs all colors of the spectrum and reflects nothing to your eye, excuse me. And so that's why you will be hotter if you go outside in a black shirt on a hot day. And if you wear a white shirt, all of the colors are being reflected back to your eye, which is why it appears bright white, and none of the colors are being absorbed. So just to know, remember, blue light and red light are the main colors that power photosynthesis, where that green light is being absorbed. So the green light is not being used in photosynthesis at all. Okay, let's move it. Oh, and then the other concept is that the longer the wavelength, the lower the energy. The shorter the wavelength, the higher the energy. So gamma rays have the shortest wavelength, the highest energy. Radio waves have the longest wavelength and the lowest energy. Alright, now let's discuss the leaf, because this is where the important stuff happens. So you know you have just a regular leaf right here. Well, you have on the epidermis, you have the stomata. The stomata are just basically pores in the leaf. You can see them good right here. That are located in the epidermis. Now if you take a cross section of this leaf, this is what you're going to come out with. This is a cross section of a leaf. It kind of resembles like a sandwich. You have your bread up top, which is and your bread on bottom, which is your epidermis. In the middle you have the meat, or your mesophyll cells. So it's just like a sandwich. The chloroplasts are actually located in the mesophyll cells, and these are the actual sites of photosynthesis. Then you have your thylakoids. Your thylakoids are actually located in your chloroplast. So if you see here, this figure does a really good job of breaking down each part of the leaf. You have your leaf right here, a cross section, epidermis, mesophyll cells. Within the mesophyll cells, you actually have your plant cells. This is a mesophyll cell. It's a specialized plant cell. And then you have your chloroplast. You can see right there the chloroplast. You can also see some mitochondria right here, Golgi apparatus, the central vacuole nucleus, some endoplasmic reticulum. But let's focus on the chloroplast. So this one gives you a close-up of the chloroplast. And like we mentioned earlier, the chloroplasts are the sites of photosynthesis. Well, it happens to be the thylakoids, these little pancake-looking things that are the actual sites of photosynthesis, of the light-dependent stage of photosynthesis, where the stroma, the liquid stuff that the thylakoids are submerged in, is the sites of the dark reaction, or the Calvin cycle. So let's just review that real quick. You have your thylakoids, that are the site of the light-dependent stage, and your stroma that are the site of the Calvin cycle or the dark phase. So let's talk about the thylakoids real quick. Okay, remember we're in the chloroplasts, and then specifically we're in the thylakoids. Well, in the thylakoids there are pigment molecules, specifically the chlorophyll. Chlorophyll is the pigment that makes the plant leaf appear green. This is the pigment that powers the photosynthesis, so it absorbs the red and blue light so it can power the photosynthesis. And so the chlorophyll is actually stored in these things that we call photosystems, which are located in the thylakoids. So let's just review that. You have a chloroplast right here. You have these pancake-looking things called thylakoids. When they stack up on top of each other, we call them a grana. Well, within the thylakoids, we have photosystems, which contain the pigments that are necessary for photosynthesis. So this is where the light-dependent stage of photosynthesis occurs. And so as you can see here, you have the sunlight that is absorbed in the pigments, which creates a energy that is used to pop that electron up to the primary electron acceptor. So we're going to discuss that here in a second. This is a light-dependent stage of reaction. Where does it occur? In the thylakoids. Remember that this occurs in the thylakoids, okay? So the sunlight, this is basically an overview of the light-dependent reaction. The sunlight is absorbed, and you can the sunlight kind of acts as like it scares the electrons away. So the sunlight comes down, the energy is absorbed, and it kind of scares these electrons up. So the electrons pop up, and they're taken to the primary electron acceptor, which kind of transports them to photosystem 1. Remember, we have photosystem 2 first, and then photosystem 1. Well, then the same thing happens again. The sunlight comes down, scares those electrons up, pops them up to the primary electron acceptor, and they're ferried over to the Calvin cycle. Well, in the process of doing this, some very important things happen. Well, first of all, we get NADPH. NADPH is another electron carrier. Remember, in cellular respiration, what two electron carriers did we deal with? NADH and FADH2. Well, in photosynthesis, we're not dealing with those electron carriers. We have our specialized electron carrier called NADPH. And we have it in two different forms, NADP plus and NADPH. Well, when it is carrying electrons, it is NADPH. Now, question, is NADPH oxidized or reduced? The key is to see that H right there, and so if you see the H right there, you see that it has gained electrons, it's carrying electrons, so it is reduced. NADP plus is oxidized. So, remember, the primary electron acceptor, the electron gets popped up to the primary electron acceptor, it takes it over to NADP pH, and that takes it to the dark reaction of the Calvin cycle. Well, in the process of this happening, you've created ATP when these electrons have fallen down the energy hill. We've created some ATP, and also we've created oxygen. If you ever wonder, remember, we breathe out carbon dioxide. That comes from the Krebs cycle and the transition step within cellular respiration. The plants are able to take that carbon dioxide in through their stomata and use that later on in the Calvin cycle, which we'll discuss here in the next couple slides. Well, you remember, we breathe in oxygen. We need oxygen. We breathe out carbon dioxide. We breathe in oxygen. The oxygen is coming from plants. So the first time that the sunlight comes in here, scares these electrons away, it has popped up two electrons to the primary electron acceptor, which takes them to photosystem 1. We'll see what happens here is that once these two electrons have become scared away and have been ferried over to photosystem 1, this photosystem 2 has lost two electrons. Well, remember, oxidation is loss. So this photosystem 2 has become strongly oxidized. Well, what happens is the water, H2O, remember, electrons travel with the H, that water splits up into its two hydrogen ions and its oxygen. And so the two hydrogen ions come over here to replace those two electrons that got scared away, and the oxygen is then released through the stomata of the plant, because the plant does not need the oxygen. The plant just wants the hydrogens. So that's how the plants are able to produce oxygen. So an overview of the light reaction. You remember, we're located in the chloroplast. That's the primary site of photosynthesis. Well, the site of the light-dependent stage, the part that requires sunlight, is the thylakoid. So that's where we're at right now. The sunlight is absorbed in these photosystem 2, then photosystem 1. The electrons have been popped up to the electron acceptors in each of the cases, and then we've released oxygen, which will be released through the stomata, which are those pores that are located in the epidermis. And the electrons have produced ATP, as well as an electron carrier, which is NADPH. And so then we go over to the stroma, which is the liquid part of the chloroplast. So this is comparable to the cytosol, but since we're inside of the chloroplast, we're going to call it stroma. Stroma is that liquid jelly substance in which the thylakoids are immersed in. So the Calvin cycle is the dark reaction, or it's also called the light independent reaction. It has all these names. You need to be familiar with all of them. Well, in the Calvin cycle, this part is called carbon fixation. So carbon fixation is the most important concept of the Calvin cycle, or the dark reaction. Carbon fixation is just incorporating a gas into a sugar. And so what you do is we have the carbon dioxide that is coming from the stomata that are located where? In the epidermis or in the mesophyll cells? The answer should be the epidermis. And so the carbon dioxide is going to become incorporated with a naturally occurring plant sugar called RUBP using the enzyme Rubisco. And so once that happens, we're going to energize this sugar we've created. We've added carbon dioxide, incorporated carbon dioxide into the naturally occurring RUBP plant sugar. That's called carbon fixation. We do that using the enzyme Rubisco. And then we're going to energize that sugar that we've created up by adding the ATP that we got from the light reaction and the electrons that we got from the light reaction, the NADPH and the ATP that we created. That's going to create a high energy sugar called G3P, and then one molecule of that G3P can go off to break down into glucose and other derivatives, while the other 5 molecules go on to start the cycle all over again. This is a continuous cycle. So let's talk about an overview of photosynthesis. So you have sunlight plus water plus carbon dioxide forms glucose and oxygen. This is basically the cellular respiration equation backwards. So sunlight plus water plus carbon dioxide yields sugar and oxygen. So the sunlight, of course, is absorbed in the light-dependent stage, which occurs in the thylakoids. So sunlight and water go into the light-dependent stage. That water splits off to form oxygen, and then we get ATP and NADPH from that goes on to the stroma, where the dark reaction or Calvin cycle occurs. The carbon dioxide is bound to the RUBP with the enzyme Rubisco, and the ATP and the NADPH from the light reaction is added to energize that sugar, create G3P that will come off and form glucose or high. Last concept that we'll discuss is photorespiration. Photorespiration occurs when the enzyme Rubisco binds with oxygen instead of carbon dioxide. So this occurs in the Calvin cycle. You know that CO2 is supposed to bind with Rubisco and bind with the RUBP. Well, what happens in photorespiration, the Rubisco binds with oxygen instead of the carbon dioxide, and so you're no longer able to, it prevents carbon fixation, and you're not able to make a high energy sugar that way. Well, the plants that have to worry about this are plants in warm environments and plants in dry environments. So they undergo another way of photosynthesis called C4 photosynthesis. So C4 photosynthesis, basically all that happens is that they use ATP to ferry that carbon dioxide to avoid, in bundle sheath cells, to avoid it from even getting a chance to bind with oxygen. And so it works. It prevents photorespiration. However, it does require ATP, and this happens in your warm environment. So your corn and sugar cane undergo this C4 photosynthesis. Well, the can photosynthesis, which is your desert animals, your desert plants, excuse me, your cactus, pineapple, your orchid, they close their stomata during the day because that is when it's the hottest. And so what they have to deal with is them drying out. Remember, the stomata are the pores located on the epidermis. And so how the cam prevents photorespiration is by closing its stomata during the day and then opening their stomata only at night. Well, and so they have less water loss. However, it's all difficult for them to bank enough CO2 because their stomata are only open at night. So it's difficult for them to bank enough CO2 to go through the Calvin cycle and produce their own sugar. But of course, the best way is the C3 photosynthesis, which is the normal photosynthesis that we discussed. But these are two other methods of photosynthesis that plants in warm environments and dry environments use. So this chart basically summarizes it that well.